Hello, everyone. Our names are Adam and Vitasta, and today we will be talking about an article on the detection of allergens by microfluidic systems. This article is by Xuan Wang and their lab group.
There are three main points to the article. Today we'll talk about ELISA, which is enzyme-linked immunosorbent assay. It is a technique used to detect and quantify allergen concentrations. The two allergens this experiment was done on are gluten and peanuts. The reason why they're doing this is because conventional ELISA requires expensive reagents and a lot of time to analyze the samples. The hope for the microfluidic version is that it'll be faster, cheaper to run, and can be potentially done on field.
To go over the ELISA system and how it works, we present this figure. We have also listed some of the key points to the ELISA process. So the way that the ELISA system works is that antigens are pushed through the channel with the hope that some of the antigens stick to the surface. After the sample is added, the targeted antibody is sent through the channel and allowed to bind to the antigen on the surface. Then markers are sent through the channels to bind to these antibodies. A positive reaction is noted by a color change in the marker. After sending the markers through the system and allowing them to react, the ELISA system can then quantify the intensity of the marker bound to the targeted antibody. This is then sent as an electric signal to a computer that then can quantify a concentration of the allergen antibody.
The allergens that this group chose to work with are gluten and peanuts. Gluten allergies only affect 0.2 to 0.5% of the US. However, 10% of the population suffers from a gluten sensitivity. This is why gluten-free options are available in restaurants and why it is important to keep allergens away from food. Peanuts and other tree nut allergies affect more than 1% of the population. Even though the affected group is relatively low, the symptoms for an allergic reaction can range from mild to life-threatening. Therefore, there is a need for a device that can quantify allergens in food on site, which is what this group is trying to provide a potential solution for.
There's a need for on-site testing for the detection and quantifying of allergens. In the laboratory, the most common method used for allergens is the ELISA test, but it uses bulky machinery and cannot be transported. Another problem is the cost of the reagents. With this in mind, these researchers propose using a microfluidic system as a solution to the cost and transportability of the ELISA test.
This figure illustrates the methodology used by the group. First, they prepare their reagents, materials, and sample. For conducting the test, the researchers created their own optical sensor and microfluidic chip capable of running the ELISA test. The next step is the antibody immobilization, where the binding of the allergen antibodies to the microfluidic wall happens. They then compare the results from the microfluidic optical sensor system to the actual ELISA test to compare result accuracy for various samples.
The microfluidic chip in this experiment was made using standard photolithography and soft lithography. The master mold was made using a silicone wafer and polydimethyl siloxane, or PDMS.
An optical sensor is used to take readings of the sample. A miniaturized optical sensor was needed to conduct these experiments at the microfluidic level. The sensor uses an LED light that measures the intensity of the marker attached to the sample. The sensor then sends an electronic signal to the computer that then displays a corresponding allergen concentration.
In this experiment, the researchers were able to develop curves that correlate the photodetector output to the concentration. This means that the ELISA test can be used to quantify the allergen concentration in a given sample. The researchers in this experiment also test the sensitivity of the microfluidic ELISA system. Sensitivity is different from specificity. Sensitivity refers to the ability of a machine to distinguish small changes in sample. Specificity, however, refers to the ability of the machine to distinguish two allergens from each other. And what researchers found is that the sensitivity of the microfluidic ELISA system is the same as the actual ELISA system.
In the future, this method for allergen quantification can be used in a multitude of industries, such as hospitality, food, or medicine. This device can be useful for governmental agencies, such as the FDA or the USDA. Future works include testing with other allergens, such as shellfish or soybeans, to see if the sensitivity is maintained.